Hello, welcome to the Decoder channel. In this video, I will show you how to run a simple correlation analysis in R. It would be a step-by-step guide from installing packages related to correlation and analysis and interpreting results using a real data set. And the data set which I'm gonna use is wheat yield data set. So it is a data set consisting of different trait values of wheat and we will check their association with yield. By the end, you will know how to visualize the relationship between variables and highlight significant ones. So let's start. So for this, we need two packages. One is called the corrplot. That is kind of basic correlation. That package is kind of basic for correlation and analysis. And the other one for calculation of p-values, that is like heatmaps. We're gonna use this package for calculation of P and R values, like P value for significance and the R value for the correlation analysis. So first thing first, if you are a beginner and if you don't know how to start, first you have to store your files in a folder in your computer, and then you have to set a directory in R. So to do this, first simply just click on these three buttons, and then you will find you can locate your folder where you have all those files related to your data, and then you select that folder and then go to this setting or this wheel entire, and then select set as a working directory, and then you are good to go. Okay, so this was like very first step. So now let's import our data. So this is our wheat yield data. So let me simply write a script where I'm gonna import this. I'm gonna use CSV file, like comma separated file, to load my data. So this wheat yield data analysis file, it has been loaded, and then we want to see what is inside the data. So we simply write head. You know what the head means. If you have watched my previous videos, then you might be familiar with. So here we have like genotypes, then we have plant height, tillers per plant, spike length, grains per spike, 1,000 kernel weight, and grain yield. So we have numeric values for all these traits. So in correlation analysis, what we actually do, we just use these numeric values and we try to correlate these traits rather than using the genotypic data. So what we want to do is we want to exclude these genotypes from our correlation data set. So we just subtract that first column from our data set. So this column would be excluded. So this command will actually exclude the first column from our data set. So then we just run this command, and then it will give us a numeric. So if you want to see what is inside this numeric data, so you need to write the numeric data set. So here we go. So we don't have the genotype column now. So we have plant height, tiller per plants, or like all the variables which we want to check the correlation analysis. Okay, so after getting this, now the fun part is, let's start the correlation coefficient and the p-values. So first thing first, we need to convert our data into matrix. So for that, what we need to do is we need to give a command to our R that convert our numeric data into a data matrix. So once we do that, and then we need a correlation matrix, and then a R matrix. So first we calculate the correlation. So this command, like this core results and this R values, we are extracting this. Here we have a correlation data and we are extracting the correlation values. You know the R values are actually the correlation values. And then we also need, we are using p-values, we're using the correlation result to extract the p-values. So here we got the p-values. Then to visualize the correlation, we will use the corrplot package. This will help you to see which variables are positively or negatively correlated. So we use the corrplot function. So here we have a small script. I will demonstrate each and everything in this script. So this corrplot is actually the plotting function. So we have corrplot, then we have core matrix as the main data set. So here we have the correlation matrix where we calculated the correlation. Then we can actually plot using different method. It would be color, shape, whatever. Like those boxes inside the corrplot may be filled with color. There may be different shapes like pie shape or what. And then we just need upper type of the correlation plot, not the both, like not in the form of square, but in form of a triangle, like upper side. And then we need color and we need some how to change the coefficient values, and then we need a p matrix, and then we need significance. Actually, we can actually for initial, what is our significant threshold, then we have like insignificance, we can use either blank or values. For this case, let's use our values and cross them, and then we can use the color of the correlation matrix. So from less correlated to highly correlated, from blue to red, and then we have the correlation plot title, and then we also can use different margins of that plot. If you want that, this is simply an optional thing like a mar function. If you want to use, then you can use, otherwise it's in really an option. You can skip that one too. So all you need to do is, after writing all that stuff, you need to click on this one and you will find a simple, nice looking correlation plot. So here you can see that each of the variable is correlated somehow with one another. And if you see these all crosses, they are showing that these correlations between plant height and the tiller plant is non-significant. And the strength of that correlation matrix, or the correlation value, is minus 0.3.1. So it means that plant height and tillers per plants are non-significantly but negatively correlated with one another. Same is the case with spike length and plant height. They are non-significantly and negatively correlated, but the strength of negativity is less compared to plant height, so it is less green. Then all this, and then here we have grain per spikes and 1000 kernel weight. They are strongly correlated. So this is the perfect correlation. One is always said that they are exactly the same values almost. So the plants or the species or the genotypes which have higher grain per spikes, they have higher 1000 kernel weight. So this is like a perfect correlation. Same is the case with the yield. The species who has like higher 1000 kernel weight, they have higher yield. So you can see the value 0.86 here and then 0.86 here. So both grain per spike and 1000 kernel weight are strongly correlated with grain yield. Anything above 0.5 means that those traits are strongly correlated with yield. So if you want, so what does this mean? So it means that if you want to increase the yield of any plant, you should increase the grains per spikes and 1000 kernel weight. So if you increase these two traits, grain yield will increase by itself. But in our data set, tillers improving plant height and tillers per plant won't increase the grain yield. Same is the case with spike length. So if you are increasing spike length, it won't increase the grain yield. This is what this correlation analysis is telling us. So if, for example, this is in current form, it is looking quite bit messy. So if you want to exclude all these crosses and these non-significant values from the plot, you just simply need to write blank here, and it will eventually discard all those non-significant values. It will remove all those non-significant values. So you will get an easy to interpret correlation plot where only significant and positive correlations are highlighted. So you can see this plot have only simple and nice looking correlation plot. So, and that's it. You have just run a full correlation analysis in R from setting up the packages to visualize significant result. If you